once again. My name is Kumar Virpan. This is the Sequencing and Introduction to HAIL Video 2, Traditional Sequencing Technology. This module is in line with the 2021 virtual workshop on StatGen methods for humans' complex traits, hosted by the Institute for Behavioral Genetics, University of Colorado Boulder. The learning objective video, or this part of the module, would be mainly to understand the overview of traditional DNA sequencing methods, which brings us to all of the wonderful sequencing methods that we've learned come across these days. So the sub-learning objectives would be to understand the basic concepts of polymerase chain reaction. Arguably, this is a StatGen workshop, which is why I won't be going too much into detail what PCRs are. Secondly, to appreciate the concepts of PCR evolving to Sanger sequencing method. Thirdly, to understand the difference between sequencing and genotyping, which sometimes people do use these two terms interchangeably, but hopefully by the end of this video, we could try to discern between the two a lot clearer. And finally, to comprehend the limitations of traditional sequencing method and why we moved on to the term next generation sequencing. There is a reason why I use the air quotes, and I will go through that in the next video. So to start with, the technological growth in genetics and genomics can be seen in this, well, somewhat exponential curve here, where on the x-axis would be the years from the 1980s to the 2010s, and the y-axis would be the number of variants that can be genotyped or detected. So in the 1980s, when we had just PCRs, and at that point even mapping genes were very difficult, we had candidate gene analysis. After the advent of PCR, 1983, and so on, is when we just started increasing the number of variants that we can detect. This then went up to hundreds, if not thousands, when we reached to linkage scans, where this is a method used in the late 1990s to early 2000s to study families where diseases were segregating in them, where we looked at patterns of linkage, and on top of that can be used as a very powerful method to look at rare segregating variants. And then when the human genome was mapped out in 2003, about 20 years, well, actually 2001, sorry, almost 20 years after PCR was discovered, is when we saw the boom of GWASs, of thousands, if not tens of thousands of variants and markers across the genome, to lead us to the genome-wide association studies that you have already learned how to run in your previous modules. And then in the year 2007, 2008, when we started having the next generation sequencing method, is where we led to really offshoot in the millions of variants and markers that could be detected, which led us to analyzing exomes and whole genome sequencing. Now, just to inform you, exomes would be coding or exonic regions that are sequenced, and whole genome would be almost the entirety of the genome that has been sequenced, and then subsequently for yourself, analyze them. So I always like starting sequencing lectures with understanding what PCRs are, because it really is amplifying DNA in vitro, in a tube. Another term that I would use for PCR would be to Xerox or photocopy multiple different specific regions on the genome or in a chromosome or just whatever genetic material you have to many copies. So the definition here, to produce many copies of a specific target segment of DNA. The main concept of this would be it's a three-step cycle of heating, cooling, replication, and repeat, which brings about to the term chain reaction and producing the identical DNA molecules. This was discovered by Kary Mullis in December 16, 1983, where, if I recall the story correctly, it was either on his Harley or his convertible with the wind flying through his hair that he found out or he thought of the idea in the car. In December 16, 1983 was when he won 1983 was when he won My apologies. 1993 was when he won the Nobel Prize for Chemistry with Michael Smith, and it was also the same year that he won the Japan Prize. Yes, I'm looking at my other screen because that's what I found from Wikipedia, because I can't remember these days. So a pictorial guide of how PCR looks like would be here. The genomic DNA, where if we want to amplify a target sequence in blue, we would have this sequence that we would amplify using something known as primers. Think of them as the start and the stop. Where do you want to start the Xeroxing and where do you want to stop the Xeroxing? So in order to get to this point, we would first need to denature at about 95 to 100 degrees Celsius. I can't really think of that in Fahrenheit. And the annealing temperature can vary depending on the primer design. It's not truly important for your knowledge right now, but that's really set in the lab that you do across multiple different parameters as well to optimize it. And finally, the extension would be mostly based on your polymerase that you're using, where most of the time the polymerase that we're using is usually a hyperthermic polymerase that usually helps or stays really well with high temperatures, because keeping in mind we're going 95 degrees going to 40 something maybe, and then the extension tends to be about 72 degrees Celsius. So this would then produce multiple copies of the target blue sequence, and by cycle 2 you have more, by cycle 8 you have 8 molecules, because 2 becomes 4 becomes 8. We're all statistical geneticists here, we can do math, and so this is the overview, and you can see it in your slide deck as well if you'd like to take it a little slower to try and just read it further. If you'd like to go through some YouTube videos, there are multiple YouTube videos out there. By all means, get in contact with me if you'd like a reference of a YouTube video that I prefer. And so this PCR led us to finally come up with the thought of we probably need to sequence out the DNA instead of just mapping it out. So based on the concept of how the primers decide where the start and the stop of these target sequences, relatively short DNA fragments therefore can be sequenced using this method called the dideoxy chain termination methods. And this method is based off the concept of PCR and how it works by design of the primers itself. So modified nucleotides called ddNTPs, or dideoxyribonucleotides, attach to synthesized DNA strands of different lengths, and this will then allow you to separate them out to then detect what each of these nucleotides are. Each type of ddNTP is tagged with a distinct fluorescent label for A, T, Gs, and C. So the dideoxy, the N itself could be either A, T, G, or C to identify the nucleotides at the end of the DNA fragment. The DNA sequence can then be read from a resulting spectrogram, or sometimes also known as a chromatogram. So how the technique works in a test tube, similar to how a PCR is with multiple different things that goes into a test tube, in this case you would have your target sequence, which usually is already an amplified target sequence, not the genomic DNA. So whatever you've PCR'd out before, you would then put it in the test tube, whatever you've PCR'd out, well, I'm using PCR as an adjective here, and you would then include a primer. As I said, this is where the concept came from. So if you want to go further back, a northern blot is really the idea of what PCRs really came about. So northern blots to PCR to now DNA sequencing, and this method is called the Sanger sequencing method. So you include a primer in order to tell your reaction and where to start, and your reaction would be made about by the DNA polymerase shown in red, and the dideoxyribonucleotides are the fluorescently tagged ddNTP, where your DDA, DDC, DDT, and DDG. You also have deoxyribonucleotides. These do not just end. And what do I mean by that? I'll show you in a few moments. So you have a mix of DNTP and ddNTP. So once you include this all and you run it in your sequencer, the sequencer in itself will then use the primer. The primer will then tell the polymerase where to start and continue sequence. And how the polymerase works is it inserts a nucleotide in at a time. So it takes randomly a nucleotide either from the DNTP or the ddNTP. If it takes the DNTP, it continues inserting one nucleotide at a time. However, when it includes a ddNTP, that's when it stops. So a reaction occurs at that point where bond breaks and stops the reaction, and then you would have just that particular length of its sequence, which is why it's really important to have just the DNA template, the DNA that's been PCR'd out instead of the genomic DNA, because it could like go anywhere and you'd have like one really long strand of the entire genomic DNA, which really can't be parsed out by a Sanger sequencer. But anyway, upon this random selection of DNTP and ddNTP, you would have randomly asserted, for instance, if the primer is TTGT, the next one would be G combined with T, because that's the complementary part. So you would have this, and then one nucleotide extra, one nucleotide extra, one nucleotide extra, one nucleotide extra, until you have the longest strand here. This is again randomly assigned, and by the end of your reaction cycle, you would have a plethora of each of these sequences being formed. And so using the traditional sequence, or using the sequencer that is available right now, it would run it through a capillary where the longest strand is scanned last and the shortest strand is scanned first, through a laser and subsequently a detector, where, well, I would say like in the 1990s, we would have a printer to print it out, and right now in the 2000s and 2010s, we would just have them in a file, usually something like an image file that you would then look at the chromatogram or the spectrogram like this. So this would be just a summary of the two slides that I had previously, and this would be how traditionally I would describe a Sanger sequencing method, by looking at the chain termination of TAGGC. And traditionally in the 1990s or 2000s, when I was doing my undergrad, is where we would run it through a gel, where you would have a tube for A, a tube for C, a tube for G, and a tube for T. So we would do different DNTPs in different tubes, and you would run it across a gel, and you literally just like look at the smallest to the largest, and you would determine T, C, G, A, G, T. This is like a huge gel, not an agarose gel if you're used to these kind of things, something known as a pageshell. Not truly important for this, but I just thought some extra information to share with you. Another thing that clinical geneticists actually look at, because Sanger sequencing method is still one of the go-to sequencing methods because it is very accurate in calling out each nucleotide. And this is what most clinicians actually look at sometimes, by comparing whether a mother or a father is contributing to the proband nucleotide. So usually when we would look at a chromatogram, you would look at how clean the peaks are, where the greens here would be the A, the G would be labeled here as G. If you do have something like this with two peaks, if you run some experiments, sometimes it's a bad thing. But in this case, because we know that humans are diploids, we know that the mother is a het for this and a heterozygote for this position, which is why you have a pink color here. And therefore, the proband in this case would be carrying the A from the mother, the A from the father, giving a homozygote for the proband and the A for that position there. So all of this really led a lot to the human reference genome being sequenced, which I love this slide that Laurie Chibnik from HSPH, someone I work with the Ginger program for Africa, that we empower people from the African continent, scientists from there, to analyze their data sets statistically. So these two papers were published in Nature and in Science, basically a day apart, reporting the human genome. I would really ask, if you want, please go and view this, just to see the small little nuances of how they sequenced out the human genome, looking at things like something like shotgun sequencing, mostly using PCR and Sanger sequencing, which is why I describe both of these technologies. And so after the human reference genome, the reference genome itself was sequenced, we then realized that this was too slow. Let's try to figure out some way to make it faster. So we used something known as genotyping chips. In the 2000s, while I was an undergrad, we used these genotyping chips on the right, which is about 2 inches by 1 inch, 2 inches long, 1 inch wide. It really doesn't matter. But the point is, on this, there are multiple probes in this blue square here, where the steps are basically getting the genomic DNA, amplifying the genomic DNA using like a PCR-like method, but randomly assigned primers, and then on the second day, basically precipitating, hybridizing it onto onto the chip, onto some probes, and then onto the chip and imaging it on day 3. And then you will have the autocall that you have gone through during your GWAS tutorial a few days ago. If you have gone through the tutorial before the video or after the video, you know what? Eventually you get there. Point is, this is how genotyping method works, where traditionally you could get about like 10,000 SNPs or markers per chip, but right now you could get up to 2.5 million markers throughout the genome on a single chip. And one of the main reasons is because there are 3 billion base pairs throughout the genome, hence the development of high-throughput genotyping using these chips, which are produced by AFI, and funnily, this cartoon here is, or this graph here is produced by Illumina. So this is also another version of just like showing you how a genotyping chip works and how the hybridization protocol works. Similar to how previously you would have different fluorescence, here you would also have different fluorescence to the probes themselves and the genomic DNA that binds. And so depending on what sequence that is binding, which is the non-bald sequence here, would be what probe would be attached onto. And this slide is thanks to Hai Liang, a colleague of mine at MGH. So sequencing methods by Sanger sequencing is great because it's still being used. It's just that it targets smaller genomic regions in a larger number of samples. The sequencing can be variable from region to region because the quality can differ. And validating results from next generation sequencing is one of the most important things that you do in the lab if you find something interesting. You could also use it to verify things like plasmid sequences, inserts of the plasmids, and mutations that you identify on plasmids. Doing HLA typing is highly accurate using Sanger sequencing compared to next-generation sequencing. Genotyping of microsatellite markers that can be done using Sanger sequencing or the high-throughput genotyping method. I noticed that we have not talked about the applications and disadvantages of high-throughput genotyping here just because you've already gone through it. And so I just wanted to mention the difference between sequencing and genotyping here. And finally, one of the final applications of Sanger sequencing methods out of the many would be to identify single disease-causing genetic variants. The disadvantages of Sanger sequencing here, which is what led us to development of NGS, would be that the sequencing of Sanger sequencing is only up to about 700 base pairs and not really much more. So we know that the human genome is 3 billion base pairs. Shotgun sequencing worked really well for doing the reference genome, which cost $1 a nucleotide or $1 a base pair. Imagine trying to get 100 whole genomes, for instance, for $1 each. That's how many billion dollars again? So it's also not great for the first 15 to 40 bases. It's just because where the primer usually binds onto. So what we usually see in the chromatogram is like really noisy 15 or 40 bases. And then you have pretty clean peaks is what we saw or how I showed you. The quality also tends to degrade after 700 or 900 bases. I would not really throw Sanger sequencing past 500 bases, to be really honest with you. And so this led us to the development of next generation sequencing methods in the mid-2000s. And these are like methods which are used at that point, like 454, which I remember in 2008, where it was used on this method called polymerase. I'm not going to go through so far. It's an emulsion type of PCR. If you notice, almost everything is a PCR-based method. And this had longer read lengths, and that's why it was thought to be really cool. However, due to the cost per megabase, that's why it was just like slowly deprecated. Solexa, which was taken over by Illumina, which is what we use right now, uses sequencing by Polymerase or bridge PCR here. The cost per megabase at 2008 was about $2, but it's gone down so much. The other three methods here, not truly important, but I just wanted to let you know that at 2008, these are what we call next generation sequencing methods. So to me, I still find it really odd when we find the Illumina sequencing method as next generation sequencing. I would just call high throughput sequencing, if you will, to follow what I think this soul-ish man. So yeah, this is a really good paper by Jason Dury, all the way back in 2008. So these are some of the machines that were used at that point, where I still remember I still wanted to get a 454 for our lab, but well, it was a Malaysian lab and we didn't really have the money to have Sanger sequencing by itself. But eventually the Luminas Alexa machines took over because it was just cheap, it was high throughput, you got reads. So this brings us to the end of the video. So by now you should be able to describe the overview of traditional DNA sequencing methods, just the overview, not even the details of which. You understand the basic concepts of PCR. You appreciate the concept of PCR evolving to Sanger sequencing methods, which then brought us to how genotyping methods work, and finally to bring us to how the development of next generation sequencing methods. Thirdly, you grasp the difference between genotyping and sequencing. And ultimately, you comprehend the limitations of traditional sequencing methods, which will bring us to our next video. This video or this session, which will be traditional sequencing method. Thank you very much. My name is Kumar Veerapan. My email address is veerapan@broadinstitute.org. Our Twitter handle is Hail Genetics, no underscore, everything small. If you have any questions, feel free to email me with your questions or send me a Slack message.